Hello students, welcome to today's lecture. We are discussing our topic molecular basis of inheritance, and in this we are discussing the gene regulation and expression, or the regulation of gene expression. In this point we have already studied that genes are switch on or switch off according to need of that organism. We have also seen that there are some enzymes which work in this relation to the gene expression, and these enzymes adapt according to the need of that organism. These are called as inducible enzyme. That process is called as induction. And in the E. coli we have seen that when the lactose is provided to a culture medium of the E. coli, then there will be formation of the three enzymes, and those enzymes are beta galactosidase, permease and transacetylase. So in this gene expression, or in the regulation of this gene expression, some point we have already discussed in our previous lecture. Now here we have to see that the model which is proposed by the scientists Jacob and Monod, who explain how the genes can be expressed or how gene can be regulated. In the last lecture, we have also seen that the activity of the gene is regulated at various levels. For example, it is regulated at the transcriptional level during the formation of the primary transcript, as well as during the processing of the mRNA, that means during the splicing stage. Second point, we have seen that when this mRNA moves from nucleus to cytoplasm, at that time also we can regulate the activity of the gene. And the last one, that at the translation level we can also activate, or we can also regulate the activity of the gene. Now here the model which is proposed by the scientist Jacob and Monod, these two scientists has made one of the model, and that model is called as the operon model, and that concept is given as the operon concept. That concept is named as the operon concept. Now what is mean by this operon concept or the operon model? So here we have to see that this operon, or this is the model which is given by the scientist Jacob and Monod. It first explains how the gene can express, or how the activity of gene is regulated. In this lecture I am going to explain whole concept of the operon, and in the next lecture, according to our textbook, we will discuss the same point. Okay, so here this operon is a unit. This operon is a unit of the gene expression. So see here what I am writing here. Operon is nothing but the unit of gene expression. Then we have to see how this model works, and before that we have to see the structure of this operon, because here we are saying that this operon is responsible for the gene expression, or it is a unit of the gene expression. Now we have to see how this operon works. So we have to see first the model which is made by this Jacob and Monod, and then after discussing the model of the operon, we will discuss how it is responsible for the gene expression. So first keep in mind that operon is nothing but a model which is made by the scientist Jacob and Monod. This model explains how genes are expressed, or how the activity of the gene is regulated by some inducers. Now we have to see the structure of this operon, and for that here I am drawing the figure of this operon. So according to Jacob and Monod, see, this is the model of the operon. First see, this Jacob and Monod propose this operon model for particularly E. coli bacteria, because in last lecture also we have seen the number of examples related to this E. coli bacteria. So here it is the model which is made by the Jacob and Monod, and in this model what is there, the different components will be found there. So this is the P, then I, again P, then there is O, and then there is a Z, Y and A. Okay, so here I have drawn the figure of this operon. P, I, P, O, Z, Y, A. So what is it? So P stands for promoter. Okay, here also another P, that means same meaning, promoter. This PP are the promoter. This I is called as the regulator gene. It is called as the regulator genes. O is stand for operator gene. It is stand for operator gene. And this Z, Y, A are called as the structural gene. These are called as the structural genes. Okay, so see here, this is the model of operon, which is mainly made by Jacob and Monod to study the gene expression in E. coli bacteria. So to study the gene expression in E. coli bacteria, this model was proposed, and here you will find that there are two sites, or we can also explain that there are three structural genes which are called as Z, Y, A. Okay, here there are three sites, P site, I site and O site. Now the three sites explain. This is the promoter site, this is the regulator site, this is the operator site, and these are the structural gene. Now here we have to see that these structural genes Z, Y, A are polycystonic structural gene, because in our previous lecture we have seen the concept of monocystonic gene and the polycystonic gene. Polycystonic genes means the genes which are responsible for formation of more than one type of the proteins are called as polycystonic gene. And when the genes are responsible for formation of one type of protein, that we call as a monocystonic gene. So these genes Z, Y, A are responsible for formation of more than one protein, so they are called as a polycystonic gene. So the activity of this polycystonic gene are controlled or regulated by two sites, and these sites are called as operator site and the promoter site. So in the operon model, there are three structural genes Z, Y, A, and the activity of these structural genes are controlled or regulated by this operator and the promoter. So it is very important point that activity of the structural genes are regulated by the promoter and the operator region. Okay, so what happens always in this E. coli bacteria when there is a source of energy, that is glucose, that we have to discuss here. But before that you should know that normal mechanism of this operon. So what happens generally in the previous lecture also we have seen that the structural genes, that is here Z, Y, A are there, and we have to see the role of each structural gene. So if we see this is Z gene, is a structural gene which is responsible for normally the synthesis of the enzyme, and name of that enzyme is beta galactosidase. Name of that enzyme is beta galactosidase. There is a Y gene which is responsible for synthesis of permease, and the A gene which is responsible for formation of an enzyme that is called as transacetylase. So these three enzymes are synthesized by the structural genes, and as we have seen that the enzymes are nothing but the protein, because in the previous lecture we have seen the function of the gene, that is the main function of gene is it is involved in the formation of the protein, it is involved in the formation of polypeptide chain. So these genes produce the enzyme is nothing but the genes are responsible for formation of this protein molecule, because chemical nature of the enzymes are protein. So these enzymes, beta galactosidase, permease and transacetylase enzymes are produced by these three structural genes. There is always question asked that what is the role of Z gene, what is the role of Y gene, what is the role of A gene. So this Z gene is responsible for formation of beta galactosidase enzyme. This Y gene is responsible for formation of permease enzyme, and this A gene is responsible for formation of the trans-acetylase enzyme. Now here we have to see that the role of each enzyme in this operon model, or role of this each enzyme in the E. coli bacteria. According to the Jacob and Monod, all these three enzymes has different role in the E. coli bacteria. So these three enzymes are involved when the lactose is present as a source of energy, because this point we have already discussed that if lactose, generally what happen in the E. coli bacteria, the general source of energy is the glucose. Always there will be glucose is present in the surrounding of E. coli bacteria. That's why this E. coli bacteria use that glucose and metabolize that glucose by using or by formation of the different enzymes. Okay, that is called as a metabolism of glucose. But if glucose is not available, and instead of that if the lactose will be provided in the culture medium, then what happen? If the lactose will be provided in the culture medium instead of glucose to the E. coli, then that E. coli switch on this operon model. So this operon model is also called as the lac operon. This operon model is also called as lac operon. Lac operon stands for lactose operon. Lac operon stands for lactose operon. What is mean is that here it is this model operates only when the lactose sugar is available as a source of energy to the E. coli bacteria. When lactose is available as a source of energy to E. coli bacteria, then and then only this model work, and this whole model is called as lac operon model because it is related to the metabolism of the lactose. So when lactose will be there in E. coli, then and then only this lac operon model switch on and it get operates. So see here, lactose is there in the culture medium. That's why the structural gene ZYA are responsible for formation of these three different types of enzymes. These structural genes are polycystonic because they are involved in more than formation of the one type of protein. When there is only one type of the protein will be formed, then we call it as a monocystonic gene. So here we have to also see the role of this each enzyme in this lactose metabolism. See here, this Y gene is responsible for formation of the permease, and the role of this permease is to entry of the lactose in a cell. Entry of lactose in a cell. So when lactose will be there, that lactose slowly enters into the cell of E. coli, and for this the enzyme permease is needed. Okay. Second is called as the second role of enzyme is beta-galactosidase. When lactose enters into the cell of the E. coli, then that lactose get broken or get hydrolyzed into glucose and galactose due to the presence of this enzyme beta-galactosidase. So lactose is broken down into glucose and galactose with the help of this beta-galactosidase enzyme. And the third enzyme, the name of that enzyme is trans-acetylase enzyme. This enzyme is responsible for transfer of acetyl group from acetyl-CoA to beta-galactosidase. So trans-acetylase function is transfer of group acetyl-CoA, transfer of group acetyl-CoA on beta-galactosidase. See here, we have written the functions of these three structural genes Z, Y, A. All the structural genes are responsible for formation of different enzymes, and each enzyme has the definite role in this lactose metabolism. So the Z gene, which is responsible for formation of beta-galactosidase enzyme, is responsible for the breaking or the hydrolysis of the lactose into the glucose and galactose. Permease enzyme, which is produced by the Y gene, is responsible for entry of the lactose into the cell. And the third enzyme is called as A enzyme, or third gene is called as A gene, which is responsible for formation of trans-acetylase enzyme, which carries out the transfer or which transfers the acetyl group, which transfer the group acetyl-CoA on the beta galactosidase. So these are the functions of the structural genes which are called as the polycystonic structure gene. So first you have to clear the concept of the operon. In this operon, structural genes are there, and the activity of the structural genes are regulated by this promoter and the operator site. You are well known about the term promoter. Promoter is the sequence which is found on the DNA. This sequence is that sequence where the enzyme RNA polymerase bind. So in this model also, the enzyme RNA polymerase, which is responsible for formation of the RNA molecule, which is responsible for the mRNA formation, are bound here. So P is the promoter site which is present at the start of the operon, and this is the site where the enzyme RNA polymerase bound. Okay. Now what is the role of this regulator gene? Here the position is very important, because for MCQ point of view also the questions are asked that where this operator is present. This operator is present in between the promoter and the structural gene. Okay. Where the regulator genes are present? Regulator genes are present in between the two promoter region. So these are the sites, that is the regulator gene present in between two promoter, operator gene present in between promoter and the structural gene. So this is the site, and now we have to see the role of this regulator gene, how this regulator gene binds to the operator region, and how it prevents the structural genes from formation of these three enzymes. Okay, so this is our first concept. We state that what is operon model, which are different genes are there, which are sites are there, how they are placed at a different location, and that is very important. So again, I am going to revise that. We have to keep in mind the spaces of the place of the each gene. Promoter at the start, there are two promoter site. In between promoter is a regulator site. Then near to promoter and the structure gene, there is operator site. There are three type of structure genes, Z, Y, A. Each structural gene produces a specific enzyme. Each specific enzyme has a specific role in the lactose metabolism in the E. coli bacteria. Now here we have to see that how this operon model gets switched on and switch off according to the chemical present in their surrounding, and that chemical is nothing but the inducer molecule, or that chemical is nothing but the small molecule which is responsible for switch on or switch off of the model of the operon, and the small molecule is called as inducer, and inducer is nothing but the lactose sugar. So here in this E. coli model we have seen the different sites, the role of different enzymes produced by this structural gene. Now here we'll see how this model works when the lactose sugar will be provided to the E. coli bacteria. At that time this model works like here we have seen the role of each genes. Okay, so here we'll see what is there in this model of the operon. First what happened, that is in presence of inducer. In presence of inducer. So here inducer means the small molecule. Inducer means the small molecule which switch on the operon molecule. Inducer is a small molecule which switch on the operon model, and this inducer molecule is nothing but the sugar that is called as the lactose sugar. So normally glucose is there, so lactose metabolism is not operated in the E. coli bacteria, but here we have Jacob and Monod provided lactose sugar in the culture which is prepared in the laboratory for this E. coli bacteria, and that time lactose metabolism get operated in this E. coli bacteria. So if this E. coli bacteria has provided the inducer lactose, then the model will be switch on, and how it works that we are going to study here. Okay, so see here first here this Z gene lactose will be provided to the E. coli bacteria. So this Y gene, which is called as which is responsible for synthesis of this permease enzyme. This permease enzymes allows the entry of lactose into the cell. and that lactose will be enter into the cell of E. coli bacteria. Later on, this Z gene provide or form the enzyme beta galactosidase. So this enzyme in the cell hydrolyze this lactose into glucose and galactose, and then third gene, that is called as A gene, forms the enzyme transacetylase. It forms the enzyme transacetylase, and this transacetylase enzyme is responsible for transfer of acetyl group to the beta galactosidase. This transacetylase enzyme transfer acetyl group to this beta galactosidase. It is one of the chemical form which is responsible for this lactose apeiron, because the role of permease you can easily understand, the role of beta galactosidase you can easily understand, but the transacetylase means it transfer the acetyl group from the acetyl-CoA. Acetyl-CoA is one of the compound. From that compound, acetyl group is transferred by this enzyme on this beta galactosidase, and then only lactose metabolism can be completed. So here the role of these three enzymes are there when they will be provided a lactose in the form of inducer to this E. coli bacteria. Then what happened here? Here you will see that this regulator gene, then what happened where this operator is there. Okay, what is the role of this operator? Operator is responsible for the formation of, or operator is responsible for the activity of the RNA polymerase, because when RNA polymerase is there, there will be formation of mRNA. mRNA will translate the protein, and there will be formation of the different types of the enzymes or proteins will be formed. So this operator is there. Okay, so here these three enzymes or three proteins will be formed when, so this is the promoter region. This promoter region forms the mRNA. From this mRNA there will be protein that is called as the repressor protein. So here we have seen the role of this, but we have to see first in absence of inducer, that is mean by in absence of lactose, when the E. coli, I am repeating the same point, that when E. coli is not using the lactose sugar, instead of that when it is using the glucose sugar, then how this lactose metabolism can be operated. So see here, first we will see, we have seen the role of this all the enzymes. Okay, here when glucose is available to this E. coli bacteria, then there will be no formation of these three enzymes, beta galactosidase, permease and transacetylase. Why? Because these enzymes are involved for the lactose metabolism only. So first we will see in absence of inducer, and then only you can understand in presence of inducer what happened in the operon model. So I will explain first in absence of inducer. So what happened that here this promoter region is first region which forms the mRNA molecule, because you know that at the promoter site there is a binding of the RNA polymerase enzyme. This RNA polymerase enzyme start the synthesis of mRNA. So in this promoter region there will be binding of the RNA polymerase, and there will be formation of the mRNA molecule. This mRNA molecule then undergoes in the process of translation, and it forms the protein, and that proteins are called as the repressor proteins. See here, that proteins are called as repressor proteins. Okay. These proteins are the repressor proteins. Now what happened? These repressor proteins then bind to this operator region. This repressor protein binds to this operator region and inhibit the activity of the operator, because the role of operator is that this operator allows the RNA polymerase to form these different types of the proteins. Okay, but here what happens, this repressor protein which is formed from this mRNA molecule binds to this operator site. That's why activity of the operator site will be blocked, and due to which this operator site cannot allow this RNA polymerase further to transcribe the mRNA, because these enzymes beta galactosidase, polymerase and transacetylase will be formed only when there will be formation of mRNA, and then mRNA will form this enzyme. So for formation of this enzymes beta galactosidase, polymerase and transacetylase, there will be first formation of mRNA, and this mRNA then form the different type of this enzyme. For first there will be mRNA formation, and then only these three enzymes will be formed. Okay, but this operator will be blocked, and that's why it do not allows the RNA polymerase to first form the mRNA and then form the three enzymes which are respectively beta galactosidase, polymerase and transacetylase, because this operator is responsible for activity of RNA polymerase. But this activity of RNA polymerase is stop here, and further formation of the enzymes will be not there because of the repressor protein, because this repressor protein are formed from the mRNA molecule. mRNA molecule will be formed by this promoter site, and this mRNA molecule binds to the operator site and it blocks the activity of operator. That's why this ZYA genes not form the mRNA. There will be not formation of these three types of the enzymes. Okay, so this is about the inducer absent. When lactose is not there, there will be no formation of the different enzymes from the ZY gene, A gene. And these enzymes, why they are not formed? Because this repressor protein is there which block the activity of this promoter site. So this is about in absence of inducer what happen in this E. coli bacteria. But when there will be inducer present, that is when there will be lactose sugar present. So in presence of inducer or in presence of lactose sugar, what happen, that here we are going to discuss and see here. Here, generally, in a normal way, this promoter site forms mRNA. mRNA then forms protein. That proteins we call as a repressor protein. Okay, but this inducer or lactose sugar is there. So here we say that this is the inducer. This inducer binds to this repressor and inactivates the repressor. Inducer bind to this repressor and inactivate this repressor protein. That's why this repressor protein cannot bind to the operator site, because here we have inactivate this repressor protein by acting of this inducer molecule on this repressor protein. That's why repressor protein is inactivated, its structure is distorted, and due to the change in the structure of the repressor protein, it cannot bind to the operator site, because structure is changed by binding of this inducer molecule to this operator. And then what happens here, the operator site is free. It allows the operator site is free. It allows the RNA polymerase to synthesize first mRNA. Okay, then from this mRNA there will be formation of these three enzymes. One is called as beta galactosidase, another one is called as permease, and then is called as a transacetylase. So here there will be formation of these three enzymes, and again the same point is that permease enzymes allows lactose into the cell. Beta galactosidase hydrolysis of that lactose into glucose and galactose, and transacetylase transfer acetyl group on the beta galactosidase, and here the normal mechanism of the lactose will be found, and the lactose metabolism can be operated here. Okay, so this is about the lactose metabolism, where you can find that how the gene is expressed or switch on when the inducer is present, that is the lactose sugar is present, and how this lac operon switch off when the sugar lactose is absent. So according to the presence of inducer, that operon gets switched on, the gene can be expressed, and according to the absence of the inducer, that model gets switched off, and those genes are not produced here. Those genes will not express here, because here in presence of inducer only the genes ZYA express, and they produce respectively enzymes beta galactosidase, permease and transacetylase. But when inducer is absent, the genes are not expressed, because at that time what happens, promoter site get blocked by this repressor protein, and that's why there will be no formation of this enzyme. That means ZYA genes are not expressed in absence of the inducer. So this model is very clearly explained how the genes can get switched on and switch off according to the chemical present in their surrounding. Because in previous lecture we have seen that E. coli adapter, one sentence is very important, E. coli adapter to their chemical environment and synthesize the important enzyme according to it, and that is here we have seen that when the lactose will be available in the surrounding medium to the E. coli, then and then only E. coli switch on some genes, and these genes form the different types of enzymes, which are nothing but the different types of the protein. But when the lactose will not be there, then this enzyme do not form. These enzymes do not form all these genes do not form all this enzyme, and that activity will be switch off. So this is the best model which explain how the gene can be expressed at some time and how this gene can be not expressed at the another time, and it totally depends on the chemical which is present in the environment. So it is very, very important that how it is operated. Now here I have given the concept of this lac operon. Okay, so see here some important things that we are going to discuss. These are very, very important, and I will revise it again, and then we will see the role of each genes. Here you should know the point is that gene regulation and gene expression, or the regulation of the gene expression. Here the best example is the E. coli bacteria. Number of MCQ are asked from this point, that is means the which model best explains the gene expression or the regulation of gene expression, then the name of that model is the operon model. Okay, this model was given by scientists Jacob and Monod. This model is mainly proposed for the bacteria E. coli bacteria, and this model, that means the operon, is a concept which clearly explains the expression of the gene or the regulation of the gene activity. This model explains where the gene can be regulated, where the gene can be expressed. According to this model, we have seen that how this operon work. Here we have observed that there is a positive control and the negative control. That means we have seen that when inducer is present, that means when the lactose sugar is present, then there will be formation of the different enzymes by the different structural genes that is called as model gate switch on. It is form different enzymes and the different enzymes play different role in that model. So it is called as a positive control of the lac operon. But when inducer is absent, lactose sugar is absent, then these genes ZYA not form any enzymes, and these enzymes are not going to form any function because their substrate, their metabolite inducer was absent, and that is called as a negative control. So wholly this lac operon depends on this inducer whether it is present or not, and according to which it works. So it is best explained by the scientist Jacob and Monod. Okay, so if you understand this model very well, then and then only you can understand how this gene can be operated. Second point is that here you should know the structure of this operon. That means this operon has the three sides. One is called as promoter side, another one is called as a regulator side, and third one is called as a operator side. You should know the role of each side. Promoter is that region of the operon where you will find that RNA polymerase enzyme will be bind there. So promoter is a region where RNA polymerase bind. Second is the I, which is also called as inhibitor because it inhibits the activity of the structure gene ZYA. So this I or inhibitor region is the regulator gene. I stand for regulator gene, which synthesize the protein, and the name of that protein is called as the repressor protein. This O is the operator side. What it operates? It operates the structural gene ZYA. When the operator region is free, then it produces the mRNA molecule. That mRNA molecule will then forms the enzymes, that is nothing but the proteins, beta-galactosidase, polymerase and transacetylase. So for formation of this or for expression of these structural genes, operator region is very important. For the expression of the structural genes, because here any protein will be formed in which way? First there will be transcription and then the translation. First there will be formation of mRNA and then there will be formation of protein. So to form this ZYA gene, these different enzymes, there will be first formation of mRNA, and then that mRNA is involved in the formation of these three enzymes. So which enzyme will be there for the formation of mRNA? RNA polymerase. So if this operator is free, it is not bound by the repressor protein, it is not inactivated by the repressor protein. At that time what happens? This operator gene allows RNA polymerase. This operator genes allows RNA polymerase to form the mRNA molecule, and this mRNA molecule will be formed from this ZYA gene. Okay, this mRNA molecule then undergoes in the process of translation, and then it forms the respective enzyme beta-galactosidase, polymerase and transacetylase. And here you will see that for formation or activity of the structure gene, the operator part is very important because it is free, and then only it allows the formation of mRNA and then formation of these three type of enzymes. So here we should know the role of this each side in the switch on or the switch off of the lac operon. So one thing is very clear: to switch on the lac operon, operator site is important, and to switch off the lac operon, operator site is important. So this operator is very important because when lac operon is switched on, operator should be free. When lac operon is switch off, operator should be bind. So this operator region decides when the operon model is switched off or when the operon model is switched on. Okay, so it is very important that operator when free, the model is switched on. When operator is blocked, the model is switched off. Okay, and just we have seen how this operator is switched off and how the operator is switched on. When the promoter produces mRNA produces the repressor protein, and when this repressor protein binds to operator, operator gets blocked, operator gets switched off. That's why there will be no mRNA formation, there will be no any types of the enzyme formation from this ZYA gene. Okay, but when this operator is free, Then what happened? This operator allows RNA polymerase to form this mRNA molecule. This mRNA molecule then forms this beta galactosidase, permease and transacetylase by the process of translation, and that model will be switch on. So this is very important, that how this structure of the genes in this operon is very important for its expression and its regulation. So here you should know that we have seen the point, and that point I have repeated number of times because number of students don't know what is meant by the gene expression in the E. coli bacteria. Gene expression occurs in every living organism, in their every cells, because how actually working of these genes is not found there, and the gene expression can be very well studied in this E. coli bacteria with the help of operon model. It is just a model which tells about how gene can express, how genes can switch on and how genes can switch off. This mechanism is same operated in every living cell, in every living organism at different times. But one example is given here, according to which we can understand the lactose metabolism in E. coli, where we can see that the activity of gene can be switched on and the activity of gene can be switched off according to the chemical present in their environment, and that is the lactose. So by this model we can clearly understand the structure of this operon and the regulation of gene expression. So in the next lecture we will see in detail how these different enzymes can be work and their role you will find there. Okay, so here we have completed the point, that is the regulation of gene expression. In the next lecture we will see that this gene regulation, their role, that means role of the operator gene, role of the promoter gene, role of the regulator gene, and again the structure genes, and how this operator is responsible for switching on and switching off the lac operon. Thank you.